Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate datasets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone. It's great to be here. I'm here with Eric Fauman, who is the Executive Director and Head of Computational Biology in the Internal Medicine Research Unit at Pfizer. If you're active on Twitter, you've almost certainly seen some of his great takes and sharing some of their latest research. He's Eric_Fauman. We're going to talk today about some of his perspectives on the role of genetics and computational biology more broadly in drug development. I've been looking forward to this conversation for a while because I've followed you on Twitter for a long time and have loved all the stuff that you put out. So appreciate you taking the time and doing this.
Yeah, my pleasure. And yeah, I've gotten a lot out of being on Twitter, and I think we'll get to talk about a lot of the connections that I've been able to make through that. And it's been a great tool for that.
Yeah, let's actually start there. I think you told me you've published 10 papers based on conversations that have started on Twitter. Tell me a little bit about that. How have those come about, and what are one or two of your favorites?
So I've been at Pfizer for 26 years. My training is in protein structure crystallography, but I got into human genetics only about 12 years ago. And I remember asking folks, what those plots are called, the Manhattan plots and all the rest. And it's fascinated me ever since. My training is in biochemistry. And so the first GWAS that I was part of, it's the Shinidol, it was a metabolite GWAS. And I realized early on that I guess my knowledge and experience with biochemistry helped me interpret all the genetic plots. I like to say that the genetic follow-up, the functional genomics that we would usually do, has been done in the prior 100 years. The example that I often start with is a GWAS of phenylalanine. You get one hit on chromosome 12, which is phenylalanine hydroxylase. The paper demonstrating that enzyme works on phenylalanine was published 53 years ago. So you don't need to purify that, do a new experiment today to prove it. So whatever the mechanism is linking the genetic variant to PAH, you can be 100% certain that the gene is PAH. So, and that got me hooked. And I think of it as like eating potato chips or doing crossword puzzles or whatever. Every time I see a genetic hit, I immediately go to try to think about, does it make sense biologically, biochemically? And that's what led me to Twitter, is that, a lot of these stories that are out there, I like to just explore for my own education, I guess, but then share that with other folks. And a series that I did for a long time that people really enjoyed was Who's That Causal Gene, where I would pick one of these hits and then walk through and just like with the PAH, sort of identify why it makes absolute sense that that's the gene. And so through that, people started reaching out to me. There was a paper that was being worked on out of the Genes and Health group where they did a knockout of the HAO1 gene and were curious about the metabolic results from that and whether those made sense. And in that case, David Von Heel put a general call out on Twitter, and somebody directed him to me. It was Claudia Lingenberg, suggested that he talk to me. And so we looked at the data and it made sense. And then I was on that paper. So that's a neat paper showing the power of metabolites to understand a drug target. Another one that I really like is Jonathan Pritchard, a grad student of his, worked on a method looking at arguing that if you have two different traits and they go in the same direction, if you have two different traits at one locus and they go in opposite directions, then the gene that you're looking at there is probably in between those two traits in some definition. When they applied that to biochemical traits, then we can actually map it to basically KEGG pathways and show that hypothesis is actually correct, that if the two traits go in the same direction, that causal gene is either upstream or downstream of both of those traits. But when they go in opposite directions, then maybe that gene is somehow in between them. And again, if you took, most ordinary disease traits, that's hard to prove, but when they are actual biochemical traits,
Right.
again, you can just go to the pathways. That's my second favorite. And the third one is a paper that just came out last week with Carol also, where I'd written now 4 years ago about one of the genes that I like to think about, histidine ammonia lyase, which is a gene in the histidine pathway. But when you do a large enough GWAS of vitamin D, it comes up as a hit for vitamin D. And we can get into details of why it would come up for vitamin D. But he was able to take that example and work up, actually use that to show how to refine Mendelian randomization in a way that can enhance your understanding of how a gene is acting on a trait. So, and we can get more into that or any of these papers.
I definitely want to come back to that one. Before we go deep into the Mendelian randomization, I'd love if you could just give people a perspective on what you do at Pfizer, and maybe the lens that we can view this through is the table of everything. When I was preparing for this, I read about the table of everything, which is an amazing name for something that you've been working on, I think, for a long time, to make human genetic data more user-friendly and part of the drug development process. Could you talk a little bit about that, how it started, what it is, and then maybe provide some of your perspective on the utility of this kind of data and approach in drug development more broadly?
Yeah, absolutely. To go back a little bit further, I've always loved biology, chemistry, math, and physics, and computers. I've been working with computers and programming for a long time. I've never actually had a computer science class, but I can code enough to get the stuff done that I need to do. And then when it gets to a certain point, then I turn it over to sort of more professional software developers. Yeah. So the table of everything comes back to the same story of the first metabolite GWAS that I worked on, where, I was trying to go through now all the loci that we had in that paper to see if I could play the same trick of using kind of known biochemistry to understand what the causal gene is. And I started talking to people about, okay, well, so what are the resources that you might use? So again, this is now going back 12 years, but things like OMIM and Orphanet, so sort of rare disease information, but also just, what genes are in a particular region, or what are the other known associations with that. And again, using my passable coding skills, I put together a little script that, given a gene, the gene symbol, would just pull back the information for that gene. But obviously, once you have it as a script, it's easy enough to just then cycle through all 20,000 genes. And so that's what I did. So the very first one was just pull back a list of all the current known protein coding genes, and then all the columns of information about that from various sources, and then just provide that as a big spreadsheet. So wanting to know, what genes have ever been associated with that trait, or what traits has this gene ever been associated with. To coming back to the earlier point, one of the things that I realized early on from looking at a lot of these metabolite loci, again, 12 years ago, there weren't a lot of positive controls of, this actually is the causal gene. I mean, that's why. But what I realized from the metabolites like PAH is that most of the time, the gene that was right there was the one that made the most sense. And so for 12 years now, I've been saying that usually the causal gene in a GWAS is the closest gene. Initially, I got a lot of pushback because there are some very well-known examples like the FTO locus where it's not the closest gene. But there weren't any really good percentages. But from that first paper, we already had about 100 genes. And from that, about 2/3 of the time, it is the closest gene. So the table of everything it's a tool for the genetics experts, but it's also a tool for the bench biologists, so who generally don't know the details of how to interpret a locus zoom plot and so forth. And so what it does is it takes every hit from the GWAS catalog, every hit from every internal GWAS, and then just maps it to a gene. And it uses just a couple of simple metrics. It uses if there's a missense variant that is in high LD with the lead SNP, even if it's not the closest gene, that's probably a good guess for it. But otherwise, it's going to give you the closest gene. And, that's going to be right about 70% of the time. So that's pretty good. And so we can roll that out to the biologist. And the biologist can very quickly say, oh, here's my gene. What are all the traits that have ever been associated with that? And then come to us with a shorter list. The reason that we put it online is because at first, again, it was just the spreadsheet that I had. And so biologists would come to me, several times a day and say, hey, could you look up this gene? Could you look up this gene?
Right.
So again, working with some very talented software developers, both internal and external, we eventually built this tool so anybody can go in and type in. And the current version, you can, right, so biologists generally just know the name of their gene and you can just put that in. But you can also put in any kind of Boolean. So you can say, oh, what are all the genes that are expressed in liver that have an association with LDL that start with the letter P or whatever you want, and narrow it down that way. And then it updates itself weekly so that as new data comes into the GWAS catalog or OMIM, that gets added so people can look those up. But again, it starts from kind of, well, this observation that, again, it's usually the closest gene. But the other thing the table of everything can do for you is you can say, show me all the genes within some distance of this gene. So the more genetically inclined folks can say, oh, maybe it's not that gene. Let me look a little bit around it. The last thing I'll say is, yes, I mean, it was called table of everything because it was sort of all the things I wanted to know about each gene and then all the genes. But it's really a misnomer because to be useful, right, biologists don't want to actually know everything because, with casting a wide enough net, anything is connected to anything. And so the key is, setting specific thresholds so that most of the time you're getting most of the useful information. Whenever I put in a new feature, I'm thinking about both the false positives and the false negatives. So what's the danger if some true information isn't in the table of everything, but also what's the danger if not true information does show up? And because we don't want biologists getting excited about something and coming to us and we have to say, oh, that's actually not true. And when we first built it, right, and the contents of the GWAS catalog have increased, I believe, 100-fold in that time. And well, let's look that up. They've increased quite a bit. Initially, we had a threshold of p-value threshold of, I think, 10 to the minus 5 or 6, because we're like, oh, we don't want to miss anything that's maybe just below.
Right.
But now with biobanks and so forth,
you get everything at that level.
That's right. So we're now using 5 times 10 to the minus 8, but even I think that's even too generous because, in UK Biobank, you've already got 2,000 traits, so you should probably be correcting for another factor of 2,000. But
how are you using this data in general in drug development? Like, what do you see? Because it sounds like you've got a hypothesis and a way of working that really combines the scale of genome-wide association studies, but also with the real deep understanding of biochemical pathways and biology. And I think what you're saying is you can't separate those two. You can't just look at the GWAS and completely divorce that from biology and hope for the best. Tell me a little bit about how all that raw material flows into insights, whether it's new targets or ways to better stratify a patient population or however it is that you think about using it.
Yeah, absolutely. My group, so we are in a group so we're embedded within the internal medicine research unit. Our group is responsible for IDEA through phase 2, but most of our work is in the early stage of target ID. And it has been wonderful seeing the evolution. Now, again, when I started at Pfizer in 1998, the human genome wasn't done yet, right? So there wasn't even such a thing as knowing the list of all genes. I've been involved, as I said, with using human genetics for the last 12 years. So even just within that, it's been wonderful seeing this evolution where, early on, many GWAS only highlighted a handful of genes that nobody had heard of before, right? I mean, TCF7L2, or okay, so what are we supposed to do with that? And so, drug discovery, I mean, there are all the papers now that show that if your target has a genetic association, it's more likely to make it through phase 2. But most targets, even today, were not first identified by genetics, right? These are all retrospective. We can go back and say, most drug discovery efforts take 10 to 12 years, but you can say now where we are today, is there genetic evidence today? There might not have been, when the project started. Yeah. And so, using genetics to help pick targets, the promise that we're making to Pfizer is, if you can find a way to get to, first in human, it's more likely to be successful there. But you have to get there, and that's the tricky part. So early on, the overlap between those genes that we might know how to prosecute and what they did and their role in biology, and the genes that were highlighted by genetics, right? That Venn diagram had no overlap. But, it's been really wonderful seeing, especially over the last few years. I mean, on the one hand, high throughput biology is giving us more information about biology, but obviously these biobanks are giving us more genetic hits. So that overlap is getting bigger in that Venn diagram. And that's really the sweet spot. So we can say, right, the biologist can say, yes, I know what assay I want to look at or what model I want to look at to validate that. And we can say, oh, that's great. And then if we get to clinical trials, it'll have a higher likelihood of success. That's kind of where we are today. Where we are rapidly moving is, and I think we can include links here. I gave a presentation as part of the Broad and GWAS catalog at the beginning of September on effector gene lists, and that's out on YouTube. And that's also kind of something that came through my exposure on social media. But so I have an hour-long presentation there where I talk about my whole philosophy of how you pick causal genes. But one of the things that I'm excited about now is that when we do have these thousands of traits, no genetic hit is in isolation. So maybe, and one of the examples I give is hits with HbA1c, which is a marker of diabetes. But if that same hit only has associations with hemoglobin and red blood cell traits and no associations with other diabetes traits, that's probably not a diabetes target. It's probably telling us something about, right? And so where I'm really trying to push people is take into account the full complement of everything that an association is highlighting. And certainly PQTLs, and we can talk more about that as well. PQTL Pfizer is part of the UKB PPP project, which generated all these wonderful protein QTLs. So having that kind of information on top of the disease information, as well as all the other sort of intermediate biomarkers or traits, helps paint a full picture of how something might be acting.
Yeah, let's talk about that. I know you're pretty bullish on the value add of PQTLs. Maybe for people who aren't familiar with them, if you could just define it quickly and then talk a little bit about why they're potentially so helpful in drug development.
Yeah. So PQTL stands for protein QTL, which is a protein quantitative trait locus. And the example that I usually start with is the IL-6 receptor. So if you just go to 10,000 people, measure the levels of the circulating soluble IL-6 receptor, you get a bunch of hits, but especially you get a hit at the IL-6R gene, which is the gene that encodes the IL-6 receptor. And we call those cis-PQTLs because the genetic variant that's driving the levels of the protein is right at that locus. If that same variant, and unfortunately I'm not in inflammation immunology, but that IL-6 receptor variant also associates with a whole bunch of autoimmune diseases and other things. So if the variant, I mean, you can take it either way. If you have a variant that associates with disease and that same variant also drives different levels of a protein, that's a good hint that that protein is involved. And not only that, but it gives you some sense of directionality that if that variant, right? So if you have the alternative allele, you have more of that protein, and you also have a higher likelihood of disease, then you probably want less of that protein. And I'll have just a quick digression. Again, I talk about this in that earlier YouTube video, and it's been a staple of a lot of presentations I've given recently. We did an analysis we compared all the metabolite QTLs that I talked about before and all the protein QTLs from this study, and we did all the co-localizations. And from that, so PAH is not one of the proteins, but at every locus where the biochemistry tells us probably this enzyme is involved, but also you have a PQTL that gives us two ways of assessing what the causal gene is, which are independent of each other. And those two methods agree 80% of the time. So from that, I take away that most biochemistry is actually happening in cells. And yet what we're measuring in blood has enough relevance that it's showing from what we see in the genetic results. But to have that, it means, again, that what we're measuring in blood is some sort of marker or shows us what's happening in the cells in a way that makes sense. And we took it even further where, if something is an enzyme and the metabolite is the substrate of that enzyme, and if you have more of the enzyme, you should have less of the substrate. But if it's the product, then if you have more of the enzyme, you should have more of the product. And that actually is also true something like 80% of the time. So it means that even if those reactions are happening in cells somewhere, the levels that you're measuring in blood are somehow relevant to what's happening inside the cells and organs. So that's why I'm excited about PQTLs. We can also, and it's different from what you would get from EQTLs, where you can do the same experiment with EQTLs and say, if I have an EQTL that co-localizes with metabolite signal, does that light up the same gene? That's only true 37% of the time, and that we have published. And again, you're better off just looking
Why
at
is that?
gene.
Why is such a big difference?
Yeah. So actually, yeah, I gave a presentation at the EBI EMBL industry program last week, and I made up a slide. I have 9 papers. Actually, I can share all 9 of those papers. 9 reasons why not to use EQTLs to pick causal genes. And they fall into 3 categories, 3 buckets. The first is, it turns out that enhancers, it's very easy to make an enhancer, right? There's so many different regulatory proteins that recognize a whole range of different types of sequences in a very degenerate way. So it turns out that almost any random sequence will affect expression somehow, somewhere. Mm. And the second, so that's sort of the one line of evidence. The second line of evidence is that it looks like, right? So if any random change to the DNA can affect expression of different genes, the cell responds by saying, well, here's a gene that I really care about, and so I'm going to have multiple regulatory elements for it. And there's a paper, Wang and Goldstein, where they showed this first, and then Pritchard had a similar paper. So it means that, right, if a gene is important to a cell, it's going to be regulated by multiple enhancers. So any one random change is not going to affect the expression of that gene very much. I should say most of this what I'm saying now is not necessarily agreed upon by everybody in the field. It's certainly my interpretation. I think this is lining up, but right, there will be people that disagree with this. But my interpretation from that is that when you get an EQTL, what you're going to get first are those genes that are only regulated by that one enhancer, which almost by definition means it's a gene that's not important to the cell. So the EQTLs that you get first are the ones that are not important to the cell. Right, or at least the transcription level of that gene is not important to the cell, probably because it's regulated by further steps downstream. And there are, of course, now several papers that look at abundance of mRNA versus abundance of protein. And there's the most
significant buffering, right? That happens. So you get a lot more
buffering, yeah. And the most, and the most I guess, understood group in there are proteins that form part of a complex. Right. So if a protein is only active as part of a complex, you don't really care how much of that one protein you have. What matters is the whole complex.
There's some limiting reactant that you really care about. Yeah.
Exactly. And the second, so that was the second set of 3 papers. The third set of 3 papers are the papers that I've been on, of trying to doing some sort of machine learning to pick causal genes. Given a lead SNP, what's the causal gene? The one that has been cited the most is the Mountjoy et al. An open approach, where again we just sort of threw in every sort of genetic measures, so distance and other things, but whether or not you include EQTLs doesn't help establish whether something is a causal gene. At the time that work was done 2 years ago, there were not a lot of PQTLs, but I bet that if you redid that analysis today with the PQTLs, and as PQTLs grow, I do think those are a much better guide to a causal gene.
Yeah. So you said something interesting a while back that I want to come back on. So the group you're part of goes from kind of target ID and deep preclinical work all the way through phase 2. There's a lot of places for there to be dropouts, failures along the way. I'm curious to you, where do you see the bottlenecks right now, and how have they shifted? Because on the one hand, we've got an explosion of genetic information that helps you get a lot of candidate targets, but we talked a lot about how tricky it is to actually pinpoint those down into the ones that have real biological mechanisms you can trust and you can drug and make sense of, and that doesn't even get into, can we actually engage the target and make it work in the patient population? Where are you seeing the biggest challenges along that really 10-year journey that can be? Maybe 5 to 7 to phase 2 if we're lucky, but I'm curious to hear about what that is today and how it's shifted.
Boy, from the genetic standpoint, again, the shift that I've seen is, I feel we did make a valiant effort at taking things that had really solid genetic support and try to turn those into targets. But what ends up happening is you spend many years trying to figure out just what's that protein, that enzyme actually doing? Maybe what's the actual relevant substrate? Where is it active? What's the most relevant assay? And And so the big shift, again, as I talked about, is that overlap of the Venn diagram of, oh, here's some genes where we have a clue what they're doing, and also they have genetic support. Because right when the Venn diagrams are separate, then right, you're trying to argue, okay, here's something again that will be successful if we ever get it to the clinic, but we're probably never going to get to the clinic, as opposed to here's a gene where, absence of genetic signal is not right, or it doesn't tell you that it's not involved. It just means that we haven't found a genetic signal, right? So the biologist is going to say, here's an assay I've already got. Here's a pathway that we already understand. Let's go here. At least I know I can get a drug. And again, so that big shift has been with these larger studies, and we can talk about, as we get to non-European populations, we're starting to find a lot more hits or whole exome sequencing helping to pinpoint what truly is the causal gene. So we have much better support for a wider range of genes. And so I think that's been the big shift. I think, sequencing, next-gen sequencing is amazing. What we haven't solved in the same way is high throughput functional characterization, right? And there are projects to try to do that. But, if you look at a graph of how many papers there are for each gene, it's a power law. So I think there's, the insulin comes up first and then it drops off very rapidly. And so still there's something like, I don't know, out of the 20,000 genes, there's probably papers on 1,000 of them, and then 19,000 others where there's hardly anything known about them.
Yeah, I mean, that's what I was going to ask, actually. Wait, like this Venn diagram goes from essentially completely separate to maybe 10% overlap or 5% if you've got 1,000 of 20,000 genes where you see a hit and you say, actually, there's deep literature that allows me to make this connection. Is that about right? Like, do you think it would be 5% and 95% of hits, it becomes almost like a PhD project to figure out what the heck's going on here?
I'm thinking about some of the projects we've done recently. I think you can, there's some tenuous connection that you can make with maybe about half of them, with about 50%. Only about 10% would I say, it's pretty well understood. That's also a double-edged sword, right? If it's well understood, then it's probably already it's already a target, or it's already well trodden. So looking for that sweet spot of, can we add some unique value here and find something that has maybe been overlooked or not studied as well.
Yeah. How do you close that gap in your view? Is it more like of these figuring out how to scale up the functional studies? Is it making sense of what's already been published through AI or other kinds of tools? Like, how much is it a lack of just, we literally don't understand it, no matter how many smart brains you could point at it, versus it's actually all out there somewhere. We need to organize it, collect it, maybe point an LLM at it.
Yeah, we can talk about LLMs because I have been playing with them a lot, trying to sort of test the limits of their knowledge. I think where we are with LLMs, as you said, is I think they're very good at summarizing what's well known, but I don't think they're at the point of being able to even approach making new discoveries. So there's certainly a role, and I think it'll be fun to talk about it more, again, to sort of summarize what's known. No, I think, again, when I think about sort of the history of biochemistry and the work that it takes to dissect the function of a particular gene, I think it's just, a lot of hard work. I am excited about things like Perturb-seq that can maybe shed light on genes that are involved in pathways where we didn't understand that before, to kind of provide some sort of guide. So there are those high throughput methods. But I think understanding, that, I for my my graduate work and my postdoc were both on X-ray crystallography of proteins. And so the protein that I worked on as a postdoc was the Yersinia phosphatase. Yersinia, so it's the Black Death parasite, and it has a phosphatase, which is unusual because the parasite itself does not have any kinases. The job of the phosphatase is to go into the host cell and basically turn everything off. It dephosphorylates everything. And I think about the years I spent looking and thinking about the cysteine at the active site that binds to the phosphate of a tyrosine phosphate. And, there's a lot of detailed work that goes into understanding one particular protein. And I don't think there's really a lot of shortcuts for that. Obviously, what I was trained in of determining the structure of a protein by X-ray crystallography that's pretty much been supplanted, I think, by
Yeah.
AI methods. But obviously that was built on 100 years of people solving crystal structures and having 100,000 crystal structures that we could train on. We're not quite there yet, I think, with biology.
This reminded me of a story that you shared. You stayed over at Linus Pauling's ranch. I assume this was when you were studying X-ray crystallography, structural biology. What would he have thought of AlphaFold 2, do you think? He died obviously 1994. So
Yeah.
but his work has definitely paved the way to what we have today, right?
Yeah, boy, oh boy. So I did my PhD with Bob Stroud at UC San Francisco, and there was a postdoc in Stroud's lab who was the grandson of Linus Pauling, and so that's how we got the invitation down. So I was there. Well, the whole group went down, and so I was there with Bob Stroud, my PI, and this person Sasha Kam, who was the grandson. So at least at the time, yeah, I mean, what an opportunity, and yet, yeah, I didn't come prepared with questions that I was going to ask him.
Yeah.
Um, I did have, besides that, I did get to hear him present 3 times on 3 completely different topics. Once was on fivefold symmetry in crystallography. Once was on the value of vitamin C, but the other time he talked about how he discovered the alpha helix and the beta strand, which is an amazing story that he was trying to figure out how to satisfy all the main chain hydrogen bonds in proteins. And because he knew there was a nitrogen and oxygen, and he got horribly sick, and his wife confined him to bed. He wasn't allowed to go to his office or the lab. But so he drew out on pieces of paper peptide chains, and then he cut them out, and then he folded them until he could see how he could satisfy all the hydrogen bonds. And the first way he did it was just fold it up into a coil, and he called that the alpha helix. And the second way was putting them side by side, so he called that beta strand. So I think he would be right. I mean, he started with fundamentals of quantum chemistry, which he invented to then understand the fundamentals of what's going into protein folding. I think he would be pretty amazed. Obviously he built on the technologies that were there. I'm sure he would build from here. I should mention David Baker was in the lab next to mine. He was a postdoc in the lab next to mine, already starting the work that he just got the Nobel Prize for. So there's kind of a continuous chain there.
Yeah.
But I mean, AlphaFold is amazing, and the engineering of protein-protein interactions, engineering new proteins, is all amazing stuff. Unfortunately, not much of what I do today involves protein structure, but I do really love when we get whole exome sequencing and can map those back onto the structure. It allows me to use my protein structure knowledge once again.
Yeah, before we transition to close out on some of your perspectives on how to make a career in an area that you're passionate about and excited about, I just did want to close the loop on AI. How are you using it today? What do you think about it? Because it sounds like you've been playing around with it a little bit, and I'm curious how people, especially who have access to the scale of data sets you have and ability to point it at, potentially some really interesting trusted sources of data to actually make sense of, versus the internet at large, where there's a lot of stuff out there.
Yeah, I guess I'll say within everything within AI and large language models, I've even within Pfizer, I've tried to carve out a niche of trying to see how far we can push the biological reasoning. I mean, we've started getting into that a little bit. The story that I told a little bit about histidine ammonia lyase, and again, that's on Twitter, and it's also in the new Tower paper, of why should this enzyme, histidine ammonia lyase, why should that affect vitamin D? It's actually not that straightforward. And so it's been fun testing different LLMs to see how they can answer that. And it turns out that GPT-4 and Claude do make most of the time they make a reasonable guess, or they make a reasonable hypothesis there of what that connection is. If you go to anything earlier, 3.5 or 3, they'll say, oh, the gene HAL is only involved in histidine, and that has nothing to do with vitamin D. It kind of gives up. If you go to even older models, they'll say, well, HAL is the computer in 2001, and why would that have anything to do with biochemistry?
Right.
And so I'm continuously testing the different LLMs with sort of long, right? So my conclusion with the LLMs today, and this is borne out by when you give them the biology GRE and some of these other standardized tests, they seem to, right, they're stuff that's well known, stuff that's in textbooks, they do pretty well at. And so I think the value today is if you're trying to get up to speed in a particular area, and again, I've been testing mostly in biology, but pretty much anything where it's in a textbook, LLMs are pretty good at bringing you up to speed because you can talk back and forth and ask as many questions as you want and have it, correct your understanding. But something where you're the expert on it, or there's only a handful of experts, you can't expect it to know that. Actually, the example that I use is I'm continuously arguing with GPT-4 about, is the closest gene usually the causal gene? So,
Right.
so most of the literature still says, oh, well, you can't, there's no guarantee. And so then I started sending it my papers and I say, well, but look at this paper. And it says, oh, maybe you're right. So
It's very agreeable, isn't it? if you wanted to reconsider.
it is. Although the other place that I'm trying to use it more is combating, right, in science or everywhere, there's confirmation bias. So once you have a hypothesis, right, it's hard to convince yourself that it's not true. And the LLMs actually are good at that. And so one thing that I've done is I'll give it, a paragraph or whatever and say, be a harsh reviewer and tell me everything that's wrong with this. And this weekend actually just blew me away in a nice way. So one of my goals since discovering that so my bias, my hypothesis is that most GWAS loci have a causal gene that makes sense some way. And so I've been trying to go through every GWAS locus. So GWAS catalog currently has 500,000 loci or lead SNPs, and I've assigned a causal gene to 100,000 of them. So I'm 20% done that way. My other goal is to have every gene be the causal gene for at least one trait, and I'm only 5% of the way through there. But a lot of these, right? So about 350 of these rationalizations of why a gene should be causal for a trait have been published in one paper or another, and I collected all the ones that I have published. I collected up on my GitHub page, just a tab delimited file, but anybody can use it. It's under, well, either Eric Fauman or EGEA for every gene ever annotated. All these have been published, so they've been in supplementary tables. But in theory, they've gone past, groups of co-authors and reviewers and editors. I mean, in reality, I'm responsible for everything that's there. I'm the one that has read through them. Nobody else has, really read through them with a critical eye, line by line. This weekend, I actually wrote a little script where I took each one of these and fed it to ChatGPT and said, tell me whether this sentence makes sense or not, and be harsh about it. One example that blew me away is one of my rationalizations. I said, I've got it written down here, auratidine is a purine nucleotide. And it said, oh, well, that's terrible because it's not purine, it's a pyrimidine, and it's not a nucleotide, it's a nucleoside. So, and it's right, it was right. So I've corrected it. But, obviously I've looked at that sentence a million times, but once you write it, it's hard to see that it's wrong. And nobody else has read it. It's been published now for 2 years and nobody has commented on it before. So I think today, one of the things that people should be using large language models for is reviewing their own work with a harsh eye, devil's advocate or sounding board or.
I use it for the same thing. I agree, and get it when I do big grant applications or something, ask it to look for consistency checks, right? If I said one thing in one place and something else that's contradictory in another place, because it's 50 pages long and I was writing page 1 3 weeks ago and page 10 a couple of days ago. So yeah, it's amazing for that. I want to get your perspective on Mendelian randomization as well. You've published a lot in space, but at the same time, I think you probably have maybe a critical eye for some. Not all Mendelian randomization papers are created equal. I wonder if you could talk a little bit about that, and also just a brief summarization for people of what the method is if they're not familiar with it would be great.
It's a wonderful concept and idea where. And the paper that I like the best is Ben Voigt and Thich Kathirison on LDL and its impact on MI. MR was already well established there, but this one really makes it very crystal clear. So the idea is in a particular gene like LDL receptor, you can have one variant that increases LDL cholesterol levels and one that decreases it. And then you can ask whether the people that have the LDL increasing variant are at increased risk of MI or decreased risk. And one way to think about it is if you could genetically engineer people to have one variant or the other, you create people, at the zygote stage that have one variant or the other, and then you come back 60 years later and you see, oh, who's had a heart attack and who hasn't. So what they show in that paper is that very clearly so your genetic contribution to your LDL cholesterol levels directly parallels your risk of having MI. And it's called Mendelian because that assortment of who gets which alleles happens early on. And so although, we always say correlation doesn't imply causation, in this case, the alleles came first. We're not saying that, oh, because you have high LDL, that changed your DNA. So, right. So generally you have instrument variables. So you have all these genetic variants that increase your LDL, and that goes on the x-axis. And then you have the outcome variables, which in this case is risk of MI, and that goes on the y-axis. And you look at that correlation. So the impact of an allele on your LDL cholesterol directly correlates to your risk of MI. And then in that same paper, they do the same thing with HDL, where they show that the same thing is not true for HDL. So you can have a genetic variants that greatly increase your or decrease your HDL that then have no effect on your risk of MI. So for drug discovery, the important thing is that, yes, HDL is a great biomarker for predicting who will have an MI, but changing your HDL, right, with having no other effect is not likely to affect your risk of MI. So that's Mendelian randomization. The problem is that, or not really the problem, but the techniques are so well established at this point, and there's so many GWAS, right? There are, I think, something like 50,000 different GWAS that have been run at this point. And as I said, you pick one GWAS and that becomes what goes on the x-axis, another one, that's what goes on the y-axis. So you have 50,000 squared possible Mendelian randomizations you could run, and you can do it with a script. And actually, I think a lot of the papers that I see, and we just talked about LLMs, I have a suspicion that it's not hard to, right? Some of these might be the result of a pipeline. You have a pipeline that picks trait A, trait B, runs the MR, and then writes the paper. And so, I guess about 6 months ago, I did a little survey on Twitter. What are people doing here in terms of these? And most people that look at these papers now just refuse to review any Mendelian randomization paper if it's strictly that and has no biological interpretation. The one that I put on Twitter that I thought was the most egregious was, right, a GWAS of who uses a particular drug. And so you can say, yes, this person has an increased risk of using that drug, but they use that as a surrogate for whether somebody was actually using the drug, was on that medication. But of course, it's not the same, and then tried to draw conclusions from that. So
right.
I think you have to stop and look. The paper that Kaur and I just put out onto medRxiv talks about, so this histidine pneumonia lyase is an example. So the histidine pneumonia lyase has associations both with histidine and vitamin D. And if you do the MR of histidine on vitamin D, you actually get no signal at all because histidine actually doesn't change your levels of vitamin D. What Kaur's insight there is that it has to do with how you're affecting histidine. And in fact, they show, what he shows in the paper then is that this gene, histidine pneumonia lyase, affects histidine and also affects vitamin D. And so rather than histidine as sort of an intermediate biomarker, it's a proxy for the activity of that enzyme. And so, right, so we can understand the biology there. We can say, yes, this enzyme, we know how active this enzyme is by looking at this biomarker over here, but that biomarker is not what's affecting the endpoint that we care about. We're using it as a proxy for how active is that enzyme. And then we can ask, does the activity of that enzyme affect the endpoint that we do care about? So you have to combine some knowledge of the biology with the interpretation.
Yeah, I like that. As we close out here, I think you've got something to offer to people around, like, actually how you decide, what questions are worth pursuing, how to spend your life, how to spend your career. It's a question that all of us have. We've got one life to live. We've typically got 30, maybe 40 good years of professional work if we're lucky. And you have a I think, simple method called TOP, talent, opportunity, and passion. I'd love if you could just talk a little bit about that and what you've learned from decisions you've made in your career.
Yeah, I go back to that all the time for myself, my group. So the TOP model says that there are these three elements for looking at any activity that you engage in. and that the ones that bring sort of the greatest sense of self-fulfillment are the ones that are at that intersection. And so again, those three elements are talent, opportunity, passion. Talent is what you're good at. Passion is the things that you get excited about. Opportunity, or also organization, really reflects, is it something that other people find valuable also? Because that gives that sense of fulfillment, that here's something that I'm good at, that I can do, that I'm excited about, and other people also find valuable. I think, we talked at the beginning, I'm excited about the stuff that I put out on Twitter that ended up in papers where people invited me to be part of their paper because they saw that I could contribute something there. So, that sort of completes that cycle. As I said, my talent in this case was I had some knowledge of biochemistry. The passion comes from this kind of crossword puzzle need to solve each of these, but that other people find that useful also is rewarding and sort of completes that. And I've used that also then in the stuff that I do in Pfizer. Now, I have to be careful because, when you first start in a role, sort of what you're expected to do is kind of laid out for you. And, when we have openings in my group, right, it's because we're like, boy, we really need somebody to do X, to work on single cell transcriptomics or whatever. So we're looking for somebody that can help with that. I was hired in 1998 for something called sequence alignment enhanced by structure. So I did a lot of that. I came up with the structural bioinformatics, given the structures we knew at the time, how do we use that to help understand our targets? That's clearly not what I'm doing today, but for the first 2 or 3 years, that's certainly what I did. But hopefully in any career, right? You start out, you show that you can do the role that you were hired for, but then you can show that, you can identify new opportunities that would be useful for the company. And that's where the O part comes in, because clearly, I have to continuously demonstrate, here's stuff that I think I can do that I'm excited about, that also will be useful to Pfizer. And the more you do that, then the more flexibility I think you get, or freedom that you get in your role. But that comes with time. Again, that's not, you're generally not hired because, you're really smart, do whatever you want. At least not not in a big company. Usually there's a role that you're being hired to fill, and you have to show that you can do that, and then you sort of earn the freedom after that.
Yeah, and so I think for people in that situation, it's really about making sure that what you picked really lines up with your talent and passion, right? If don't pick something, especially if it's prescriptively laid out, you're going to do sequence alignment or whatever it was that you're doing. You probably had some innate talent and passion for that. Otherwise, you wouldn't have absolutely nailed it and then got on to the next thing, right?
Yeah, I think that's true. And the last thing I'll say here is the other thing that I'm a big proponent of is what's called, I call it the brand of you, of right, knowing what it is that you're excited about and making sure other people know that too, because you don't necessarily know all the opportunities that might come your way. But if you make sure that people know that you're known for something, again, like the HAO1 paper, somebody else on Twitter directed David Von Heel to talk to me because I'd sort of planted a flag that, hey, I like talking about metabolite GWAS. And I think that happens within the company also. You make sure that this is what you're known for, either by, internal presentations that you give or the projects that you choose to take on or not take on. But that's very important for ensuring that you can end up at the middle of that TOP Venn diagram.
Yeah, perfect. I think it's great advice. Thank you for sharing it. And just more generally, I really enjoyed this conversation. Like I said at the start, I've been wanting to talk to you for a little while because I followed you on Twitter for a long time. I think this actually ties it together in a nice loop, because what you were doing on Twitter is you're just letting your talent and passion shine and putting a banner out. And often the opportunity finds you if you're authentic and you're putting out there what you're interested in. So yeah, thank you for and it's a huge net positive to the community, right? It's so much everything you put out is free, and people like me can benefit from reading it or listening to it. So thank you for sharing all that with us.
I will mention I've kind of shifted to not on a daily basis, but at the end of each month out on LinkedIn, I survey. Again, I have this sort of insane goal of trying to annotate every locus from GWAS catalog. So at the end of each month, I look at all the new GWAS hits that have come out, and then I write about that on LinkedIn of, here are the top new hits in the categories of protein QTLs, metabolite QTLs, and disease traits. And I always find something interesting.
Yes, that's awesome. So people can follow you on LinkedIn, and add you on there, and maybe somebody who wants to help you crack away at that. Is it not growing faster than you can annotate it? Hopefully not.
And maybe we have another podcast about that. Obviously, a lot of every time somebody does a diabetes GWAS, you're going to get TCF7L2 and so on. Right. So a lot of those genes come up again and again. So I've only got 1,200 causal genes that explain the 100,000 lead loci that I've got so far. So so although yes, the number of lead SNPs in the GWAS catalog is continuing to grow exponentially, but
they're saturating a small set of genes, basically.
Yeah, I think so.
Yeah, interesting. That tells us something.
I think what we're saturating is white European genetics, which is just a tiny slice of all human genetics. So it's another whole topic we didn't even get into, but I think there's obviously tremendous opportunity as we finally move beyond that we will find lots of new interesting genetics. And there are the startings of that in lots of different countries and in populations. But
that's a great call. That's a really good call. I'm looking forward to, now that the All of Us data is much more widely available, I'm looking forward to more coming out there. And there are a number of large scale biobanks outside of the US and European clusters that have produced them historically. So it seems like we're making progress there, but it couldn't come fast enough.
Whole nother topic. We could certainly fill a whole other hour.
Definitely. I will take you up on that. Next year we'll do a deep dive into this. Thank you. And thanks everyone, as always, for listening. We'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.